Good morning or good afternoon, everybody. Still waiting for today's speaker. Here we go, perfect. So it's a tremendous honor and a pleasure to continue our Global IMMUNOTALK series with another excellent speaker, Monika Wolkers. I'd like to briefly introduce Monika to you. She's originally from Germany. She has lived for a very long time. Actually, I don't know for how long, how many years you lived in the Netherlands. So you are somewhat, I think, more Dutch than German. And scientifically, you have always been super excited about T cells. You started your scientific career in Amsterdam in the lab of Ton Schumacher. There she studied antigen presentation due to T cells during infections and tumors. She then transitioned to the US, was a postdoc with Steven Schoenberger and Doug Green. And here she focused on T cell programming and cellular and molecular requirements for forming long-term stable memory T cells. In 2010, you returned to the Netherlands, started your own lab, which is located at the National Blood Bank of the Netherlands. The institute is called SunQueen. I hope I pronounced this correctly, in Amsterdam. There she continues with T cells. You study molecular mechanisms that dictate the stem effector T cell function. You investigate how post-transcriptional events regulate or how nodes regulate and define T cell function, and specifically a focus on regulation of T cell function by RNA binding proteins. And using molecular biology and immunological approaches, you demonstrated how and which RNA binding proteins are responsible for fine-tuning T cell responses. And recently, you and your team have focused on fundamental rules that define the overall protein expression in T cells. And all that is not enough. More recently, you became interested, as we all do, in cancer immunotherapy, and you're trying to bridge how sort of fundamental research and drive innovations in fundamental research to improve immunotherapy against non-small lung cell cancer and pediatric solid tumors. Now, I could mention other things. I could mention prizes that you have. I should mention that you recently was awarded an ERC grant, which is, of course, a huge success in Europe, the most prestigious grant you can get in Europe. And but here I'll stop, and I'm very happy to have you. We're looking forward to your talk, which is entitled Translational Control of T cell Function and How We Can Modify It. But as usual, we want to start with our brief question. And what I would like to ask you is if you can share a moment of your career when you doubted that you're on the right track. And the question is, how did you navigate this uncertainty? So thank you very much, Monika, for being here.
And thank you, Dietmar, for having me here, because it's really a fantastic and a great honor to be part of the Global IMMUNOTALKs. So how I want to answer this question is actually the following, telling about what happened when I came back as a postdoc from the States with a grant that allowed me to set up a reporter mouse for a specific gene that I wanted to study. And I was halfway, but then I learned that a very renowned lab in the United States already had made the mouse, which was already bad, but which was even worse was there was no way of seeing this gene, and the reporter mouse didn't work. So I was a little bit shattered and didn't really know how to respond. And someone then, I thought like my scientific career was done, but someone told me, you know what, this is the best what could happen to you. You have 1.5 years on your grant. This gives you the chance to do whatever you want. So I sat down, read all literature that I could get on, because I knew I wanted to still work on T cells, and looked for questions that to my opinion were unanswered. And that is actually how I built up my career on post-transcriptional regulation in T cells.
Well, I think it's a very nice example of how sort of unexpected things drive you into something new. And
Yes.
I think what the key message is, there's always something to discover next, right? One thing is finished, but the beauty of science is we always have things and new things we can discover, and we just have to be creative and think about things. Okay, perfect. Now with this, I'd like you to share your slides with you, and we look forward to see your presentation.
Can you see my slides?
Looks good, yeah.
Yes, excellent. Oh, so I will just make myself smaller, I hope. Oh, wait. I will need to undo this one. And well, Anyway, I will just start. So let me please indeed thank you again for having me here and allowing me to tell you about translational control of T cell function and how we can modify it. So as you all know, T cells are obviously a very important immune cell type against pathogens and tumors. And I drew here the line of the fate of a T cell from naive effector, memory, and recall responses, just to highlight that we have different phases of T cells. We have, of course, T cells that are very important during the primary infections to clear pathogens and also actually to clear malignant or pre-malignant cells. We have a second phase, which is the memory phase, which really gives us long-term protection, because once we get reinfected by an infection, the T cells now can protect us from really becoming sick. One thing that I really would like to highlight of this is that throughout the life cycle of a T cell, it requires continuous signaling. And what do I mean by that? So when we have quiescent cells, so naive cells or memory cells, you will know that T cells get either continuous but very low signaling through the T cell receptor or through cytokine receptors, which allows them to have a little bit of signaling through mTOR or PI3 kinase, and by that allowing a bit of transcription and also a bit of selective translation that allows them to get the right survival signals and to divide every now and then. Once the T cells become activated, however, the scene changes, and now we have very strong signaling through the T cell receptor and co-stimulation. And now the key drivers of T cell activity, the mTOR signaling and MYC translation, now really drive the T cells to produce tremendous amounts of effector molecules. And that is indeed what really drives the efficacy of T cells during the effector phase. One thing that they really make a lot are cytokines and perforins and granzymes, which, when I say actually T cells, I mean CD8 T cells. They will kill the tumor cells. And we have focused a lot on cytokines. And why did we do so? Because it's clear from many studies that the production of cytokines is important in the T cell function against tumors, as exemplified here by a study from Philippe Bousseau, where we clearly can see that the efficacy of CAR T cells is diminished once they're not able to produce interferon gamma anymore. However, these cytokines are also highly toxic, as you know, and when we have too much of a production, like in juvenile rheumatoid arthritis, we get immunopathology. And when they make too little, we get tumor outgrowth, or we have chronic infections. So this means that the cytokine production must be very tightly regulated. So how is this achieved? When T cells exert their effector function, they have many molecular or mechanistics how they can drive this. So it can be transcription regulation, it can be the metabolism that defines the T cell effector function, the amount of signaling, and also the RNA fate and translation. I will today primarily focus on the signaling and the translation, and on the mRNA fate. So why do I think this is really important to study? And that is because the protein abundance of T cells is not only regulated by transcriptional regulation, so how much RNA is actually transcribed from the DNA, it's also by post-transcriptional regulation. And that really is a key factor that defines how much from a messenger RNA is in the end actually becoming a protein. And why do I think this is important? Because the messenger RNA abundance in this case, a human T cell, compared to the protein abundance, doesn't really correlate that great. And this is a very conserved feature, because also in other cell types, even down to yeast, we will find always this correlation factor. And how does it look like when you see an RNA abundance of, for instance, about 100? I will exemplify this here for two very important transcription factors for T cells, which is TBET and STAT1. TBET and STAT1 really have the same levels of messenger RNA, but once you look at the protein level, you can clearly appreciate that more than 150-fold higher levels for STAT1 are measured as opposed to TBET. So this really clearly tells us that protein levels do not nicely correlate with messenger RNA, and that this is highly regulated. How this is regulated, for understanding that, we need to zoom into the messenger RNA. I depicted here messenger RNA with the 5' untranslated region, the coding region, and the 3' untranslated region. And how protein output is regulated occurs on many different levels. We have translation control, so how much initiation factors bind, and how RNA binding proteins control, block, or drive the translation. We have ribosomes that define the translation efficiency and the speed, and how much of a protein is actually a result as the output. The levels of RNA are also very tightly regulated, either by stabilization or by degradation of the molecules. Then there is post-transcriptional modifications like M6A that really can occur everywhere, and that also again define all these features, so stabilization and translation, but also splicing, subcellular localization is regulated, and all these features together define the actual protein output. So let me give you an example of how this actually looks, and I will zoom into the 3 cytokines that are the most important ones to our understanding for CD8 T cells, which is TNF, interferon gamma, and IL-2. Why do I zoom into these ones? Because we have found that they have an individual use of regulation of gene expression. While, as you probably know, TNF is very rapidly produced by T cells and then declines also rapidly after a T cell activation, interferon gamma has also a rapid production, even though less so, but will be produced for a very long time, while IL-2 really takes a long time before it's produced. And what we found is that for this very first burst of cytokine production, translation control is very important. Then in the second phase, the de novo transcription defines the magnitude of the response. And then in addition to that, RNA stability defines also the magnitude, but also the length of the cytokine production. And what we found is that actually all these 3 cytokines have an individual use of these features. While TNF is primarily driven by translation control, interferon gamma depends on actually all of these 3 features. And for IL-2, we really first need de novo synthesis before actually protein can be produced. Let me then zoom into memory cells. And for memory cells, we actually have also seen, and this holds true in this case here shown for mouse memory cells and for any type of memory, but this is also true for human memory T cells. There's always preformed RNA for TNF and interferon gamma, and not for IL-2. But even though they have messenger RNA, they don't produce protein. So what we found, and this is all published, so I will cut the story very short. What we found is that there's A-rich elements in the 3' untranslated region that are really the key factors that block the translation of these preformed messenger RNAs. And we also have learned that actually this preformed messenger RNA at the same time allows for these rapid recall responses that we have seen in the first 2 hours. Effector T cells are indeed key in producing these cytokines and other effector molecules. However, once they're continuously triggered, we see that they lose their effector function. But what we saw is that this messenger RNA, and for instance, for interferon gamma, we actually still find substantial levels of messenger RNA, yet there is no protein production. And again, to see whether these A-rich elements were important for this, we made use of a mouse line that has a germline deletion of these A-rich elements in the interferon gamma messenger RNA. And just by changing this part, so nothing on the coding sequence or anything else, this sufficed to make these T cells much more potent against the tumor, as you can see here from the tumor outgrowth. These cells indeed were producing also much more interferon gamma, and we could show also that this actually was at least in part driven by higher RNA stability. We also found that this is conserved in human T cells. So I hope this made clear that post-transcriptional regulation is really a key factor to define how much cytokine a given cell produces. So how does this occur? So I've shown you this picture before, and we have actually what I won't show you the data because again, it's published. We found that one of the RNA binding proteins that we identified, ZFP36L2, is the key factor in blocking the translation in the memory T cells. It is actually released at the moment that the cell is activated, allowing for this rapid translation to occur. What we also found is in a later study that this RNA binding protein regulates the stability of messenger RNA once T cells are activated for a longer time. So it has a dual role. But there's many other RNA binding proteins. The current estimate is more than 2,000 RNA binding proteins. So what about those ones? How are they interacting with messenger RNA? And here we again focused on the 3 cytokine 3' UTRs. So what Branka did was taking these 3' UTRs and asking which RNA binding proteins act with cytokine RNA. And how does this interaction change once a T cell changes its state and is now activated? So for answering these questions, Branka did a pull-down assay. So what she did is using primary human T cells that she activated, let rest and expand for a couple of days, and these cells were either activated or not, and then she made whole cell lysates of it. This was used as a source of the RNA binding proteins because now she came with streptavidin aptamers to which the 3' untranslated regions of these 3 cytokines were fused, and the empty aptamer was used as control. She used that as a bait to isolate these RNA binding proteins, and this was then subjected to mass spectrometry analysis. And from this picture, you can appreciate that all the 3 cytokines have different patterns of the interaction of these RNA binding proteins, as one would expect. And I will highlight one of the RNA binding, one of the 3' UTRs, where you can hopefully appreciate also the changes of this interaction of 3' UTRs, because here you see all the RNA binding proteins that interact on resting, and here on are derived from activated T cells. And you can see here RNA binding proteins that specifically interact with the 3' UTR once the T cell is activated, these ones when it's resting. There's a third one, a third category where we see that the interaction is increased. And this protein, ZFP36L1, is actually a sister protein from the one that I showed you before. It's also zinc finger RNA protein and also binds to A elements. So what happens when we deplete this RNA binding protein from primary human T cells? If we do that and look at early response rates, not so much actually, so we don't see much of an effect. But once you look at later stages, when in the control treated cells, we see clearly that the responses are going down. This is not the case for ZFP36L1 depleted cells, where we clearly see that the production of cytokines still here shown for TNF and interferon gamma, but the same holds true for IL-2. So the interesting part was that this RNA binding protein is actually increased and highly expressed in the most dysfunctional tumor infiltrating lymphocytes in a melanoma study. So this was a single cell RNA sequencing study. And therefore, we also thought it would be interesting to look whether this depletion of this RNA binding protein would alleviate the block of activity in the in tumor infiltrating T cells. So we went into the mouse and we depleted this RNA binding protein in OT1 T cells and gave these cells to B16 overbearing mice. And when we just looked directly ex vivo for the tumor infiltrating T cells, you can again appreciate that the deficiency of ZFP36L1 results in increased cytokine production and also to a somewhat improved control of the tumor outgrowth. So this actually tells us that RNA binding proteins can contribute to T cell function in vivo. So I told you a part of ZFP36L1, but also of ZFP36L2. So what Norden actually wanted to know whether there's a division of labor of these 2 proteins. So what he first did is just looking about the expression of these RNA binding proteins. And he found that actually, indeed, as you would expect from the data, the ZFP36L1 is really expressed during the earlier time points of T cell activation, while ZFP36L2 is expressed later. So what happens if we now delete both of these RNA binding proteins because they bind to the same region? So what we did was taking indeed a mouse we had at hand, which comes from Martin Turner's lab, which is a CD4 free mouse with a depletion of ZFP36L2. And in these T cells, we CRISPRed out ZFP36L1. So that made that we had wild type cells, we had ZFP36L1 cells, and so single knockouts and double knockouts. And this was important because we already know from other studies that in naive T cells, if you deplete more than one of these partners, they don't give rise to good immune responses. But with this, we could actually look what happens in effector T cells. So you can here clearly see again that we could beautifully knock out ZFP36L1. What we did then is actually taking these mice that are either wild type or single or double knockout, and we exposed them to B16 over target cells in vitro. And we did that after 24 hours. We plated them again on B16 over seeded cells, and then again. And then at different time points, we measured the amount of cytokine production of these cells. And this sort of allowed us to reflect what you would see in vivo with a continuous antigen exposure. And this is how it normally looks like for the wild type cells, which really stop producing interferon gamma upon this continuous production, which was partly relieved once we looked at the two single knockouts. However, it came to us to a quite big surprise when we looked at the double knockout, which just produced tremendous amounts of cytokines. Really telling us that these proteins act in concert to suppress the cytokine production continuously. We also learned that there's much more messenger RNA, which is completely stabilized at this late stage. However, when we looked in vivo, we did not find a better control of tumor outgrowth. And that was actually because the cells die more when we have a double depletion, they proliferate less. And also when they're unactivated, we found block NG0. So this clearly tells us that one has to be careful in fine-tuning these or in altering the expression of these RNA binding proteins. So to conclude this first part, I hope I could convince you that RNA binding proteins define the magnitude and duration of protein expression in T cells at least, but also in other cell types. The activity of these RNA binding proteins is context and time dependent. And we have learned that modifying RNA binding protein expression can be used to alter T cell responses, but careful design for doing so is key. So let me move to the second part of my talk, which is the signaling part. And I would like to specifically focus on the mTOR signaling during T cell responses. We know it's really a key factor in driving the translation in activated T cells. That has been shown. It has also been shown to be important for cytokines. So we wanted to know how this occurs. So to do so, we again went back to the human T cells and we activated them either with or without mTOR-specific inhibitor taurin-1, which actually inhibits both, but we also use rapamycin. I won't show you the data, which specifically inhibits mTOR1, and that showed us exactly the same data. So this is work from Anouk in the lab, and what she wanted to first see is whether indeed also in the human T cells we find this this effect of mTOR inhibition on cytokine production, and that's indeed the case here as shown for IL-2. It's a little bit less so for interferon gamma, but clearly also found for the TNF production. So what do we know from mTOR signaling? We know that it promotes protein synthesis through the 5' untranslated region. So once T cells are activated, we have an active mTOR, which now phosphorylates two translation inhibitors, 4-EBP1, which then results in the release of translation initiation factors. And the other event that occurs is that it can phosphorylate LARP, which is bound to the 5' UTR and also by that blocks the translation events to occur. Because we worked on 3' untranslated regions, actually I should also mention that this is a selective target mRNAs. Because we worked on the 3' untranslated region, Anouk wanted to know what the role of this is. And she was triggered by the following finding we had before, which is that indeed we knew that the 3' untranslated region is really key in driving the cytokine, the protein output. So when we look here at reporter constructs with containing the 3' UTR of interferon gamma or IL-2 or TNF, you can, and this is at 0 hour, we have quite a low expression of the GFP protein. But once you activate T cells, you can clearly see that this block is relieved and that we get a strong induction of this protein. So Anouk wanted to know what happens once you block taurin in this case. And indeed, when she now looked at the 3' UTRs, she indeed found that when compared to the DMSO control in blue, there was a clear suppression of the increased protein production for TNF and even more so for IL-2 and interferon gamma. And because taurin can block general translation as well, we also looked at two controls, which was the GFP alone control, where we hardly saw a shift, and the granzyme B 3' untranslation, we also hardly found this shift. So this clearly tells us that these 3 cytokines are subjective to a 3' mediated regulation through mTOR. So how does this work? We know that LARP binds to so-called top motifs that are very CU rich. Intriguingly, these top-like motifs also are present in TNF, IL-2, and interferon gamma. So the most logical thing for us was making fragments and then look at whether this it is sufficient to have the regions with the CTT motifs to convey this regulatory effects. However, that was not the case. So when we looked at the full-length interferon gamma 3' UTR, we saw this again, this great shift in block of protein induction, which we found still being present with the fragment 1, but it was completely lost with the fragment 2 that contained the CTT motif. And that is again shown here for interferon gamma with higher, with all the experiments that we did. And the same holds true for IL-2. It's really the fragments 1 and 3 that relieve this, that drive this block, and the same holds true for TNF. So what is in these other fragments? Actually, one of the sequences that are in there are these A-rich elements. So what we then did was point mutating from the full-length 3' UTRs, either the A-rich elements or the CTT motifs into non-functional motifs. And what we then found is that indeed, when we compare to the wild-type 3' UTR, the CTT mutants did not result in a relief of this taurin inhibition, but the A-rich elements did. And again, here you will find it quantified. So what binds to these A-rich elements in the 3' UTR? So we went back to this pull-down assay that Branka previously had established in our lab, and we now actually made, the cell lysates from reactivated T cells that were either treated or not with taurin, and we subjected aptamers that contained either the wild-type 3' UTR of IL-2 or the A-rich mutant of the very same 3' UTR. So what did we find? So number one, we obviously found the classical A-rich element binders, like as the ones that I already told you before, ZFP36L1 and L2 that you will find here highlighted, and its third family member, ZFP36. But we also found that these interactions are not sensitive to taurin. So what is sensitive to taurin? We found 13 different proteins. Some of them we found lost their interaction once we blocked taurin, and others actually increased it. And most importantly, all of them were independent, required the A-rich elements in the sequence to interact. So to further look into what these RNA binding proteins were doing, we chose for 5 of them, 4 that we found lost the interaction, and 1 that gained the interaction with the A-rich element upon taurin treatment. And we gene edited again in primary human T cells, and we made use again of the GFP construct containing the wild type 3' UTR, because we know that taurin can have many effects, and also RNA binding proteins can have many effects. We only wanted to look at the direct effects on the 3' UTR. So when we now made all these mutants and we looked at, upon 12 hours of activation, how the levels of protein expression was, we found that the first one didn't do much, but all the other 4 that I show you here show clearly lowered protein production when when the ARID element binding protein is lacking. In addition to that, 2 of them were also taurin insensitive, or rendered the cells taurin insensitive. That was TCP1 and DDX21. TCP1 is a known mTOR target, and we did not follow up on it because the cells really suffered very badly due to the knockout. So we decided to continue with the DDX21 knockout cells. So what did we further learn from this? We learned that, that number one, when we looked in the DDX21 knockout cells and compared it to the control cells, where we find the taurin sensitivity sort of halved the expression of the GFP. In the DDX21 knockout, we found the same limited expression, really resembling the taurin effect in the wild type. And lastly, another control that we really needed to do is looking whether the A-rich element sequence is very important. And therefore, we also included a control with the A-rich element mutant. And we did not see an additional effect, whether you had the single mutant or the double mutant. So is DDX21 now a direct target of mTOR signaling? Actually, yes, because we know from literature that DDX21 is phosphorylated by mTOR signaling. It's actually one of the highest target of mTOR, of mTOR1, as shown in Jurkat cell, but also in activated primary mouse T cells. And you can appreciate here that there's really an increase of binding upon T cell stimulation. And in addition to it, and quite importantly, when we look at raptor knockouts, which is a key component of the mTOR1 complex, this phosphorylation is completely lost. Lastly, we also wanted to know whether DDX21 can also interact with the other 2 cytokines. And indeed, what we found is that not only for IL-2, 3' UTR, we find a binding upon RNA immunoprecipitation, but the same holds true for TNF and for interferon gamma. So to conclude the second part of the talk, I hope I could convince you that mTOR signaling can promote the protein translation also through 3' untranslated regions. We found that for cytokines, at least at DDX21, that can convey this translational control, and that this interaction is, I actually can't read it because my thing is in front of it, but that this is really driving the protein production. So I told you a lot about protein regulation for the 3 cytokines, and we really like working with them because they have such a different way of control. However, we also wanted to get a better gist on the general translation control in T cells. So which genes actually alter their translation profile upon T cell activation? To study this, Branka and Nicolina joined forces and analyzed changes of ribosome occupancy of messenger RNA in activated T cells. So how did they do that? They took resting T cells, made again a whole cell lysate, and then fractionated the cells depending on the amount of ribosomes that were bound to the messenger RNA. And this was then subjected to proteomics and RNA sequencing. And when one looks at the RNA that is pulled down, one can, and looks at each of the fraction, one can really distinguish the free messenger RNA with the monosome-bound RNA and the light and heavy polysome fractions. And what Nicolina then did was performing a pattern clustering to define the distribution of a given messenger RNA between these fractions. So what you can see here then is that each line is a messenger RNA, and it's then indeed determined how much you will find in which fraction. So you can do that then for all genes that we, or all messenger RNAs that we sequenced, and then you get different features. So you get one that where you will find that the majority will be in the free, so there's no ribosomes bound, hence also no RNA protein production. You have some where there's really this monosome peak, others that are sort of all over the way, and sometimes a little bit more peak in the polysome. But the more you go to the right, you can appreciate that there's more ribosome interactions. So how does this distribution now change for these RNAs when a T cell is activated? And that's what you can see here. We really get a substantial redistribution of ribosome occupancy. Some of the messenger RNAs will stay in the same cluster, as you can see, for instance, here for the purple one on the top, that you are a non-ribosome bound or ribosome free, and you will also be ribosome free once you activate a T cell. But others are really changing into clusters that are now bound to ribosomes and will now also be subjected to translation, while others actually that are highly in the highest translated fraction now go down. And we're using currently these type of analyses to define how the RNAs are distributed in the polysome fractions, and whether we can get clues of how RNA binding proteins drive the ribosomal occupancies. But what we also want to understand is which parameters define the translation control of these cells. And to this, we actually took a step back. We know from many studies that 3' UTRs and their length influences the messenger RNA and protein abundance. The longer they are, the less of a protein output you will have. There's many other features like the CG content or sequence conservation or codon usage or RNA modifications and so forth, that all of them have been shown to contribute to the protein abundance in a cell. However, these features are not isolated and they influence each other. So what we wondered is whether we can integrate all these features to decipher the rules that define the protein abundance in cells. And so what Benoit did is using a machine learning approach to decipher the code of gene expression. So what he did was taking all RNAs on all variants that he could find in data, and he chopped them up in 5' UTR, the coding region, and 3' UTR, and he included in this model RNA binding motifs, RNA binding protein motifs, messenger RNA modification sites, GC percentage, length, sequence conservation, codon usage, putative PT, post-translational modification sites, and so forth. And he used them in a model which I will not allude to, but you can read on it in this preprint. He used that to predict the protein abundance. As input, we used many cells, but I will start off with the cell lines, the K562, HeLa, and HEX cells, where we could really predict more than 60% just by using these sequence features, the protein abundance. And I hope you can appreciate it from this graph, where you can see on the x-axis the predicted protein abundance, and on the y-axis the measured protein abundance. And I think we are doing quite decently in predicting the proteins. For the other primary immune cells that were isolated from human PBMCs, we also, in most cases, reached 40 to 45%, but we weren't very much content about it. So we also were wondering whether we can improve the prediction of primary cells. And that we did by including the RNA measurements. So when we now include the sequence features that are on the messenger RNA together with the amount of messenger RNA you will find in the cell, we can increase this prediction capacity by, on average, by 11-fold. So going from 40-something to above 50% of the proteins that we can accurately predict just based on the sequence feature and the RNA measurements. And there is some cool stuff that we actually find with it. So one is that this usage of these sequence features is clearly cell type specific. So when we use these features and now look in the cells what type of features they like most, we actually find a very nice clustering for the cell lines, B cells, T cells, and NK cells, quite close to T cells actually, and then here dendritic cells. So this really tells us that it's very cell type specific, what is an important feature and how the messenger RNA correlates with the protein output. So the beauty of SONAR is indeed that it's audible and that it uncovers the relative contribution of these features. So here as an example, you will really see the features in a 5' UTR, CDS, 3' UTR, and also post-translational modification sites that can now be interrogated. And it also, so that's number one. Number two, what we also know now is indeed the relative contribution of the regions of the messenger RNA, and not so surprisingly, the CDS is really the prime contributor to the output of the protein, since it's the coding region, and this has been reported before to be the most important regions. But second and third immediately are the untranslated regions. One thing that SONAR cannot do is to inform us about the directionality. So meaning that when a feature is highly important, whether it's actually a positive predictor for protein abundance or a negative predictor for protein abundance. So we wanted to see whether we can experimentally test the contribution of these sequences. And that's what Caspar did. So what we did was making a 3' untranslated region that was completely devoid of sequences that we knew would be important for prediction. And we inserted 6 times a motive of interest into this custom 3' UTR, which we then transduced either in hex cells or CD4 or CD8 cells, and then we FACS sorted low or high cells, so to know which ones are correlated with high or with low protein abundance. And by deep sequencing, we could then look at which sequences would be important for this. And what we found is that a high AG content of the 3' UTR really was a negative predictor of the protein output, and the CT is actually a positive predictor. Another important part is that these features are not only changing between different cell types, but they also do upon T cell activation. So to look at it, we made use of a data set that we got from Roger Geiger, where he showed T cell activation and where he measured not only the transcriptome level at these time points, but he also measured the de novo protein synthesis by pulse labeling, and the full proteomics, so the final output, the abundance that you would find at all these time points. And again, we got some surprises. So while I already told you that the CDS is really important for the protein abundance, and as one would expect also for translation rate, given the codon codon usage and so forth, this region is actually also very important for the messenger RNA abundance. Another thing that we also found is when we looked now at the top 100 sequence features for each region is indeed when we look now at the activation status from 0 to 6 to 24 hours for the RNA data, that we clearly see indeed again that it's most important in the CDS, but that there's a lot of changes in the importance upon a T cell, upon activation of the T cells. So telling us that not all sequences are equally important at a different cell state. And this also holds true for this translation rate and for the protein output. So indeed, again, sequences are time dependent, whether they're very important, yes or not. So can we now exploit the gained knowledge to boost protein expression? So for doing that, we first went back to the GFP models and indeed introduced sequences that SONAR predicted to be very important. And when we looked in T cells that expressed them, this would be the control vector that contained just scrambled parts in the sequences. I think in the summary down here, you can appreciate that the yellow one has a little bit of an improvement of expression. The red one is a really good sequence feature to drive protein production, while these other two, the blue and the purple one, really suppress the protein production. I told you that there is a cell type specificity for these cells, and that's also what we see for these single versions. So again, CD8 and CD4 cells behave very similarly. So with red and orange being boosting protein output, while the other two are blocking it. This is not the case for K562. Here, actually, the yellow one becomes more important. For HeLa cells, we see again a difference. Now the yellow one is also not important. And for HEX cells, again, the blue and purple is not important, but those ones are important. Again, indeed showing the cell type specificity of these features. So this is GFP, but how about a more relevant protein? So we went to a neoantigen-specific T-cell receptor that we got from Ton Schumacher, which is recognizing a mutated CDK4 in a melanoma line. So what we did again was making use of these sequences, and we added either the scrambled 3' UTR or just left the one that was before it, or we introduced the sequence features into this scrambled 3' UTR. And again, we found that the yellow and the red one increased the protein production of the T-cell receptor, while the other two, the blue and the purple one, again blocked it. This was not because the RNA was higher. The RNA levels were actually equal. And which you can also appreciate here when you look at the fold change of messenger RNA versus the protein fold change. It's really translation control that defines whether what the actual levels of the 3' UTR of the T-cell receptor is. Does it matter? So we again made use of the same cells, and we now looked in an in-Q site for the killing of the T cells that expressed the different 3' untranslated regions. And this is the untransduced cells, this is the control cells, the AU-rich or the purple and the blue ones containing, and there we don't find any effect whatsoever. But once we use the other ones that have a higher T-cell receptor expression, they now also kill the tumor cells better than the other ones. So with this, I'm at the end of the talk, and in the last part, I hope I could convince you that the machine learning pipeline SONAR informs us on sequence features that predict the protein abundance. This feature importance is cell type specific and also depends on the activation status of a cell. SONAR can be used to modify protein expression, and I didn't show it here, but it can also be used to modify general protein production by cell lines. With that, I'm at my end, and would like to thank this fantastic team who did all the work. I already mentioned everyone along the side. We have brilliant collaborators, and of course, I also want to thank my funding. Thank you very much.
Well, thank you, Monika, for the fascinating data and a very excellent talk. I'm sure there's a lot of questions that many people like to ask, but as usually, we have to refer to Twitter or X, how it's now called, for any questions. So please make use of that opportunity to ask many questions to Monika. With that, I'd like to finish today's session with highlighting our speaker who comes next week. So it's another originally German speaker who also lived long in the Netherlands. So we'll continue as we had it today. So what I mean is our next speaker is going to be Carmen Gerlach, who is in Stockholm. And with that, thanks again, Monika. And I wish everybody a remaining day, very nice day. And see you hopefully next week. Bye-bye.
Thank you. Bye.